好，嚟到2013年嘅第6條長題目啦，就係有關於蒸騰作用嘅。咁今次呢個裝置咧就係幫我哋去揾翻呢個有葉嘅植物啊嘅莖部咧，佢嘅失水嘅速度、蒸騰作用嘅速度啦。咁成個實驗咧，其實就係喺我哋書本學嘅氣泡蒸騰計。咁當然你會發現題目俾你嘅裝置同書本嘅裝置咧係有2大個分別嘅。第一就係呢個微細管 capillary tube 嘅形狀啦，呢個咧就直條嘅，呢個就係 S 型啦。但係入面啦其實都有氣泡嘅，所以佢哋呢2個嘅氣泡蒸騰計嘅運作原理係一模一樣。咁唯獨較為特別啲嘅就係呢個蓄水器啦。咁呢個蓄水器有咩咁重要呢？就係當我開呢個擰掣，當啲水放落嚟嘅時候呢，就幫我哋將呢個氣泡歸零啦，推翻去一個0 cm 嘅位置，或者將佢推翻去一個原本嘅位置啦。咁 Part A 就問啦，當我哋做呢個實驗要有呢個裝置嘅時候啦，點解呢棵植物嘅莖部嘅下半部嘅部分，點解要擺水去剪佢呢？咁其實啦，正喺書本咧已經提咗我哋啦，就係防止氣泡進入木質部，從而就令到棵植物吸唔到水。所以成條題目咧，其實就係考緊我哋木質部係攞嚟吸水。相應嘅技巧咧，就係當我哋去裝置呢個蒸騰計嘅時候，有啲咩嘅預防措施啦。咁所以答案咧亦都呼之欲出啦，就係避免木質部被氣泡鎖去阻隔，從而就令到棵植物吸唔到水啦。而 Part B 就問啦，當我哋運用呢一個裝置去量度失水速度嘅時候，究竟我哋要作出咩嘅假設 assumption 呢？咁所以 Part B 咧，其實就係考緊我哋一棵植物，佢之所以能夠吸水，就係源自於一道力叫做蒸騰牽引力，而蒸騰牽引力係由蒸騰作用咧所導致嘅。而第二個考我哋嘅概念咧，就係有關於一棵植物保留住嘅水，其實係點樣去量度出嚟呢？就係透過佢嘅吸水減佢嘅失水啦。相對應嘅技巧呢，就係有關於做實驗嘅時候有啲咩嘅假設。咁我哋先講第一個概念啦，就係有關於失水同埋吸水嘅。一棵植物失水嘅時候就會產生蒸騰牽引力，從而咧就有一串水珠咧就喺木質部向上拉扯，從而就幫棵植物咧去吸水嘅。所以成個概念就係講緊一棵植物係先失水後吸水。睇翻成個實驗，佢聲稱就話係幫我哋量度植物嘅蒸騰作用嘅速率，但係個氣泡之所以喐，係因為棵植物吸水。所以成個氣泡蒸騰計實在幫我哋量度緊嘅係棵植物吸水嘅速度，而唔係失水嘅速度。所以成個實驗係一個好致命嘅破綻。咁究竟我哋可以有啲咩嘅假設，去令到呢個 set up 係真係可以幫我哋量度到呢個 leafy shoots 嘅失水速度呢？我哋就係假設翻呢棵植物係唔會儲存任何嘅水分喺個植物入面嘅。所以啦，呢個 water retention 等於 water uptake 減 water loss 嘅呢條數式咧，保留水嘅份量係0啊。而我哋將呢個失水掉去左手邊，就令到成條數式就會變成吸水等於失水啦。所以喺 Part B 我哋要假設嘅就係失水速度係等於吸水嘅速度，又或者吸翻嚟嘅水就淨係攞嚟失咗去嘅啫。咁當然呢兩句你會發現啦，其實冇可能噶，梁 Sir。但係一棵植物係需要保留水分俾佢棵植物嘅正常運作噶嘛，攞嚟做光合作用都需要水啦。植物要令到啲細胞硬脹，從而產生一個支撐能力嘅，都要用到水噶。所以呢度咧就提翻大家，如果大家係想溫下有關於一棵植物吸水失水嘅話咧，就快啲睇翻呢一段吸水理財學啦。梁 Sir 就將一棵植物嘅吸水失水同埋經濟學嘅原理冚埋一齊。嚟到 Part C 咧，又係一啲較為直白嘅題目啦。今次就要我哋解釋下，如果成個裝置隔籬我擺把風扇，而將呢把風扇開咗佢，咁呢棵植物嘅蒸騰作用嘅速率有啲咩嘅改變呢？咁所以成個 Part B 咧就係考我哋啦。究竟一啲環境因素係點樣去影響得到蒸騰作用嘅速率？今次講緊嘅就係開風扇，所以講緊嘅環境因素就係風速或者空氣流動嘅速度啦。而我哋咧係需要解釋翻點解空氣流動嘅速度係會影響到蒸騰作用嘅速率嘅。所以成個作答框架咧，第一分你就要講翻究竟蒸騰作用嘅速率有咩嘅改變，1分。然後再解釋翻呢個生物嘅現象啦，所以就有1+3嘅呢個配搭。其實我哋書本咧正正已經學過呢個情況噶啦，風速增加 失水速度增加。原因何在啊？就係因為當氣流加快嘅時候咧，就會將呢棵植物周遭嘅水蒸氣就會吹走，或者你可以講翻風咧，就可以將氣孔附近嘅水蒸氣吹走都可以。咁吹走咗啲水蒸氣啦，又要咩咁特別咧？就會令到個氣室同埋大氣之間嘅水蒸氣嘅濃度階梯就會變得斜咗。呢個答法咧，我係中意一啲嘅。咁啊，因為書本咧佢淨係話啦，保持一個斜嘅濃度階梯。咁但係我想你哋演繹嘅就係佢係斜啲啊，因為而家係講緊開咗把風扇啊嘛。咁即係話你應該係比較下開風扇同埋熄風扇之間嘅情況。咁而家開咗風扇咧，水蒸氣嘅濃度階梯應該係斜咗嘅。咁所以有翻少少比較式嘅字眼咧係好啲嘅。所以啦，水蒸氣嘅擴散速度咧係會快咗嘅。咁啊擴散出去大氣啦。咁我哋最尾嗰句咧，我又想你學翻書喎，因為淨係講擴散去到個大氣咧，又係爭少少嘅。擴散係由一個地方去到另一個地方啊嘛，咁所以啦，你應該係講下個 water vapor 水蒸氣係由個葉嘅氣室係擴散出到去個大氣嗰處，呢件事係發生得快咗。其實呢一 part 係反映咗你答題夠唔夠精準嘅。而呢個題目啦，有啲咩嘅變奏呢？就要我哋描述或者解釋一下喺唔同嘅環境情況底下，對於呢個蒸騰作用究竟有啲咩嘅影響啦。而第二款嘅變奏呢，就要我哋描述或者解釋一下，當一棵植物面對一個太強勁嘅失水啦，咁究竟佢個氣孔會有啲咩嘅變化，變大定變細呢？同埋點解呢？到 part D 咧，正正就係題型嘅變化啦。我哋今次咧就講下氣孔咯。咁你見到啦 A、B、C 唔好諗啦，即刻做咗 labeling 先啦。A 咧就係葉嘅表皮細胞。B 咧就係保衛細胞，而 C 咧就係我哋嘅氣孔。今次得一個，所以佢係 stoma，而 stomata 咧係眾數嚟嘅。嗱，今次佢冇問你 A、B、C 係乜嘢，下次可能問你嘅。最陰陰濕濕嘅咧就係如果你將 C 答咗做 stomata 咧，唔好意思啦，真係錯嘅，因為你應該答嘅係單數。而 part D 嘅第一 part 就講啦，比較下 cell A 同 cell B 佢哋兩者嘅分別啦。但係今次呢個分別咧係講緊細胞嘅結構嘅分別，有啲咩係唔可以講呢？就係比較佢哋個細胞嘅大細或者形狀啦。而由於我哋要比較嘅關係，我哋相對應嘅 critical skills 咧就係完整比較。當 cell A 係咁或者佢有啲乜嘅時候，但係 cell B 就係咁或者佢有啲乜啦。所以 cell B 咧保衛細胞佢係有葉綠體，但係 cell A 係冇嘅。而保衛細胞嘅細胞壁佢係不平均嘅厚薄。但係 cell A 咧就係平均嘅厚薄啦。而去到第二 part 咧，就問下我哋啦 ，size of C 即係氣孔嘅大細啦。喺夜晚嘅時候會有啲咩嘅變化咧？而我哋就要解釋一下呢個變化嘅功能重要性，其實就係我成日講嘅適應性特徵，係佢呢個結構究竟同佢功能有啲咩嘅關係啦。咁所以成條題目咧就係考緊我哋光度嘅變化對於氣孔嘅大細會有啲咩嘅改變，而氣孔有咩嘅功能咧，就係同失水同埋氣體交換有關係啦。我哋之前喺書本都已經有學過噶啦。當日頭猛嘅時候咧，氣孔係會打開，能夠吸多啲嘅二氧化碳進行光合作用，但係同一時間咧，失水嘅速度亦都會快咗。但係喺夜晚嘅時候啦，佢對於光合作用嘅需求係零啊，佢係唔需要去吸二氧化碳嘅。而去到夜晚嘅時候咧，個氣孔係會閂埋咗，所以就會導致到咧失少咗水啦。所以第二部分咧，第一分我哋就要答嘅就係氣孔喺夜晚嘅時候佢係會變細噶，而原因就係當一棵植物喺夜晚嘅時候佢係唔會進行光合作用嘅，所以呢棵植物對於空氣交換嘅需求咧亦都冇咁大啦。咁你話喂，梁 Sir 佢仍然都需要氧氣進行呼吸作用嘅喎？係啊，我冇講過氣體交換嘅需求係零啊，我只係減少咗，因為減少咗棵植物對於二氧化碳嘅吸收嘅需要啦。好，又嚟到一點出發嘅時間啦。今次嘅題目咧就用蒸騰計作為切入點，考我哋嘅咧就係氣泡蒸騰計。下次未可以問我哋重量蒸騰計啦，係咪？ 今次咧，我哋就利用氣泡蒸騰計去進行科學探究，去研究下一棵植物吸水同埋失水嘅關係。題目亦都要考到我哋，當我哋預備一個實驗裝置嘅時候，有啲咩嘅預防措施同埋假設呢？而第二條路徑就係問我哋有關於蒸騰作用啦，風速、光度對於蒸騰作用嘅速度有啲咩嘅影響呢？仲有冇其他環境因素都會影響咗蒸騰作用嘅速度呢？今日又嚟到好東西要分享嘅時間啦。之前梁 Sir 咧就拍咗2段片，係有關於重量蒸騰計點樣計數嘅。咁嗰啲數呢 MC 又可以問你，長題都可以問你。如果你唔係咁識計呢啲數嘅，快手睇咗呢2段片佢啦。咁呢條題目啦，做啲咩嘅變奏呢？今次呢條題目就係利用氣泡蒸騰計去探究一下啦。當我哋改變風速嘅時候，呢棵植物嘅失水速度係會點樣變化呢？自不然就可以問下你3個 V 啦，係咪？咁第一個要講啦，自不然係自變項風速啦。但係今次梁 Sir 咧想講多一樣嘢，就係我哋如何控制個風速嚟啦。開風扇、熄風扇，或者改變個風扇同個植物嘅距離，擺近啲咪大風啲咯，擺遠啲咪細風啲咯，或者調校個風扇嘅速度啦，唔係淨係開與閂咁簡單。第二個啦，因變項自不然就係講緊失水速度啦。咁你話唔係，梁 Sir 頭先你未講嘅，第二個裝置係有破綻嘅，佢係量度緊一棵植物嘅吸水速度。但係你要記住啊，我哋做咗假設噶嘛，我哋係假設咗一棵植物嘅失水速度就等於佢嘅吸水速度，個 dV 咧仍然係失水速度。我哋要留意嘅就係我哋如何量度呢？我哋係量度緊個氣泡移動嘅速度。例如啦，佢30分鐘入面究竟行咗幾遠呢？個單位咧應該係 cm per minute， 每1分鐘行咗幾多 cm， 從而呢個就係速度啦嘛，距離除時間啊嘛，係咪？而去到最後尾就係控制變項，有啲咩嘢我哋喺唔同 set up 都要保持一樣呢？例如啦，今次我哋研究緊風速，咁溫度啦、相對濕度啦、光度啦。呢棵植物佢有幾多塊葉，或者啲葉嘅大細，我哋盡可能都要揾啲一模一樣嘅啦，甚至乎用翻同一棵植物啦，係咪？作為破解 SBA 嘅實驗性題目呢，梁 Sir 係會詳盡解說俾大家聽嘅，希望呢段片幫到大家。213 question 6 is about transpiration. We are going to use this setup to determine the transpiration rate, and this setup is what we learn in the textbook, which is the bubble potometer. Of course, you can see that there are two main differences. First of all is the shape of the capillary tube. On the exam paper, it is a strict capillary tube, and in the textbook, it is an S-shaped capillary tube. But one common thing we can still find is the air bubbles. So that's why the mechanism behind these two setups is the same. The second difference is the water reservoir. What's the function of it? Once we open the tape, the water will go down and then move the bubbles, go back to the 0 position or the original position and be ready for the next round of the experiment. And obviously, this question is checking the water absorption and transpiration concepts. So in part A, in setting up this experiment, the lower end of the leafy shoot should be cut underwater. What is the reason behind that? In the textbook, it tells you the answer already. This prevents air bubbles from entering the xylem vessels and blocking the water uptake. So the first concept checking is the water absorption by the xylem vessels, and the corresponding critical skills is about the precaution of using potometer. So for part A, it's very direct answer. Avoid the blockage of xylem vessels by the air bubbles formed during cutting, and it will prevent the water absorption. Part B, give one assumption for using this setup to measure the transpiration rate. So for part B is checking the concept about water absorption along the xylem is caused by transpiration pool, and the transpiration pool is generated by the transpiration. And the second concept is about the water retention in the plant is equal to water uptake minus water loss. And the corresponding critical skills is to make assumption for the experiment. And this question, the critical concept is the whole story of transpiration and water absorption. Firstly, there is water loss from the leafy shoot, and it will generate a transpiration pool, an upward pulling force in the xylem vessels for the plants to absorb water. Therefore, water intake follows water loss. We try to organize the idea. This setup is claimed that the potometer is used to measure the rate of transpiration, which is the water loss. However, the air bubbles movement is caused by the water absorption by the leafy shoot. Therefore, the potometer is actually measuring the rate of water absorption by the leafy shoot. So you can see that actually the question is lying. The setup cannot directly measure the transpiration rate. So any assumption can be made in order to make this setup to help us to measure the transpiration rate. It can be assumed that the leafy shoot does not store any water in the plant's body. Therefore, the water retention is 0. And then we transfer the water loss to the left hand side. And then water uptake is equal to water loss. Therefore, we can make the assumption that the rate of transpiration is same as the rate of water absorption. Or we can say that the water absorbed is used for transpiration only. So that's why the plant will not retain any water for photosynthesis, for providing support by turgidity. And Mr. Leung has already made a video about transpiration. How can we link biology and economics together? You can watch the video about the cash flow in the plants. Let's move on to part C. It's very straightforward question. Explain how the transpiration rate will change if the fan placed near the shoot is switched on. So in this question is checking the concept about the factor affecting the rate of transpiration of the leafy shoot. It's because we turn on the fan, therefore the wind speed will increase, or the air current, the air movement will increase. And then we need to demonstrate the mechanism which the higher wind speed affects the rate of transpiration of the leafy shoot. So for the scaffolding, the first mark we need to state how does the transpiration rate change. For the following 3 marks, we need to explain this biological phenomenon. And we have already learned this concept from the textbook. First of all, the transpiration rate will increase. And what's the reason behind that? Because the air current will blow away the water vapor around the leafy shoot. Or you can say that the wind will blow away the water vapor around the stomata. And the concentration gradient of the water vapor between the atmosphere and the airspace in the leaves becomes steeper. By comparing this statement with the statement in the textbook, I prefer you really talk about steeper, but not just mention maintaining a steep concentration gradient of water vapor. I want you to do some comparison of the concentration gradient of the water vapor when the fan is turned on and turned off. So we use the superior terms. And then the water vapor will diffuse out from the airspace to the atmosphere at a faster rate. But for this statement, I prefer you write something from the textbook because I want you to tell me the direction of the water vapor diffusion. It should be from the airspace to the atmosphere. Please tell me the diffusion, it takes place from where to where. Any possible question variation? First type, to describe or explain the effect of different environmental factors on the rate of transpiration. For example, light intensity, relative humidity, and so on. And for the second type of variation is to describe or explain the size of stomata when there is excessive water loss from the leaves by transpiration. And for part D, is the question about the stomata. So in this picture, it shows the appearance of the surface of a leaf during daytime. So we can see the cells A, B, and the structure C. So for the good practice, do the labeling first. Cell A is the epidermal cell, cell B is the gut cell, cell C is the stoma, not the stomata. Stomata is the plural form. You can see that during the daytime, the stoma is opened. In part A, in terms of the subcellular structure, state 2 differences between cell A and cell B. So in this question, the concept checking is about the subcellular structure of the epidermal cells and the gut cells. So you cannot compare the size or shape of the cells, and the corresponding critical skills is complete comparisons. Cell A is something something or has something something, but cell B is something something or has something something. You need to tell me cell A and cell B at the same time. Therefore, this question is very straightforward. Gut cell contains chloroplast, but cell A does not. The second difference is the cell wall of cell B has uneven thickness. However, the cell wall of the epidermal cell is evenly thickened. Part 2. What will happen to the size of the stoma at night? And discuss the functional significance of this change. So you may ask that, Mr. Leung, what is functional significance? In fact, it is the alternative term of adaptive features. You need to link the structure and the functions together. So for the concept checking of part D2 is about the effect of light intensity on the size of, and then we need to link the change of the size of the stoma to the rate of transpiration and gas exchange. And that's the concept we learned from the textbook before. During the daytime, the stoma is open, and in darkness, the stomata are closed. Therefore, for the first part of the question, the size of the stoma at night will be reduced. But what is the importance of this change? It helps the plants to reduce the transpiration or water loss. Meanwhile, in darkness, there is no photosynthesis. Therefore, the need of gas exchange decreases in the absence of photosynthesis. The plants, they only need to open a small gap for the oxygen uptake, but they don't need a larger stoma to absorb more carbon dioxide for the photosynthesis. So let's talk about the curriculum mapping in question 6. This question, it starts from the potometer, and this question, it asks you about the bubble potometer, and next time it may ask you the waste potometer. And because we use the bubble potometer for the scientific investigation, this question, it keeps checking the concept about the relationship between water loss and water uptake. And it also checks us the critical skills about the precaution and assumption. For the second part of this question is about transpiration rate. How do the wind speed and light intensity affect the transpiration rate? And any other environmental factor will affect the transpiration rate. Mr. Leung has made 2 videos about the examination skills about the calculation in the waste potometer. So if you are not that familiar with the calculation in biology, so watch these 2 videos immediately. Any possible question variation inspired from this question. So in this question, we are using the potometer to determine the transpiration rate of a leafy shoot, different condition. For example, the wind speed. As a scientific investigation, of course, we can ask you about the 3 variables, right? So it's time to do some revision together. First of all, for the independent variable, of course, it is the wind speed. In this question, I would like to add something more. How can we manipulate the IV? We can turn on or off the fans. We can change the distance between the fan and the plant. When we move the fan closer to the plant, the wind speed is higher. And when we move the fan far away from the plant, so the wind speed will be lower. And for the dependent variable, of course, we are measuring the rate of water loss. You may ask that, no, Mr. Leung, you say that the bubble potometer, in fact, it is measuring the rate of water uptake. Yes, I agree. But remember that we have made the assumption. We assume that the rate of transpiration is equal to the water uptake. Therefore, we can still claim that the setup is helping us to measure the rate of transpiration. Okay, so when we are measuring the rate of water loss, in fact, what do we really measuring? We are measuring the rate of air bubble movement, which is the distance moved in 30 minutes, or it depends on how long do you do the experiment. So in this case, distance moved in 30 minutes, so the unit will be cm per minute. And the concept is just the speed is equal to distance divided by the time. And for the control variable, what do we need to keep constant in different setup? So for example, the temperature, relative humidity, light intensity, or the number of size of the leaves in the leafy shoot. Or even we can use the same leafy shoot for the experiment. I hope that this question about the scientific investigation or SBA can help you to do the revision a lot.